Reproduction is based on heredity. Content. Every living thing contains a substance described as genetic material. Except for certain viruses, the material is composed of the nucleic acid DNA. DNA has an underlying linear structure in segments called genes, products of which direct the metabolic activities of cells. An organism DNA, this array of genes, is organized into structures called chromosomes, which serve as vehicles for transmitting genetic information. So there are two processes, mitosis and meiosis. Both of these happen in nucleated cells. So both of these processes, they run similar, but the ending result will be different. Because meiosis will create 4 cells with half of the genetics. Mitosis will create only 2 cells, which are exactly the same as the mother cell. So mitosis is a portion of cell cycle during which hereditary components partition equally to daughter cells. Meiosis only happens in sex cells. Meiosis is essential in the transmission of genetic information from organism to the offspring. Nucleus. Living organisms are categorized into 2 major groups depending on whether they have or not. So prokaryotes are organisms with no nucleus. Eukaryotes are organisms with a nucleus. So nucleus will be this structure that houses the DNA within a membrane. So during mitosis and meiosis, chromatin fibers coil and condense into chromosomes. Also present in nucleus will be nucleolus. Nucleolus is an amorphous component. Relevant RNA are synthesized, and where the initial stages of ribosomal assembly occur. The portion of DNA that encode RNA are referred as the nucleolus organizer region or the norm. Prokaryotic organisms do not have a nuclear envelope or membranous organelles, but there are 2 of them, such as eubacteria and protobacteria. The eubacteria, like Escherichia coli, the genetic material is long circular DNA molecules condensed in a small region called nucleoid. Part of the DNA might be attached to the membrane, but in general, the nucleoid extends to a large part of the cell. So the DNA might be compacted, but it doesn't really coil similar to the ones in the eukaryotes. So in the eukaryotes, all of the chromosomes are formed from the coiling of the DNA. But for the bacteria, the chromosomes does not form at all because the DNA does not coil to that. For the picture here, you can see that the bacteria has formed into 2 daughter cells by cell division. You call it the binary fission. So in the binary fission, you can see that the nuclear regions has turned into 2 of the same size and same shape. So both of these nuclear regions contain the bacterial chromosomes. Prokaryotic cells do not have an existing nucleolus, but do contain genes that specify rRNA molecules. So the chromosomes, they are visible only during mitosis and meiosis. If they are not dividing, the genetic material would unfold to form a chromatin. In chromatin, they are formless, you cannot see anything at all. It's just like a dark region. But for chromosomes, when you look at them carefully, you can see that they have their own shape, their own size. Each chromosome would have a region called centromere, where it forms the waist of the chromosome, meaning that there's an area which is a little bit narrow. We call it the centromere. Picture 3, it shows chromosomes with different centromere placements. If it's in the middle, it is metacentric. If it is between middle and the end, it is submetacentric. Close to the end is acro, and at the end is telocentric. The shorter arm is called the P arm. P is the short form for petite, which is the French word for small. The other arm, we call it the Q arm, because it is the next letter in the alphabet. So the ability of organism to reproduce distinguishes living things and non-living matter. So whenever something is living, it is able to reproduce or propagate. So every cell has to undergo the cell division as part of its cycle. So you have cell divisions in prokaryotes and eukaryotes. In prokaryotes, there is only one type of cell division, which is binary fission. Eukaryotes, you have mitosis and meiosis. Mitosis is for growth, development, and repair. And some of the animals or organisms that can do asexual reproduction will do mitosis only. So usually the cells will be identical to the parent cell. And also it occurs in the somatic cells or body cells. Meiosis is the one that is concerning sexual reproduction. So it yields genetically different cells with half the number of chromosomes. It occurs in specific reproductive cells and yield gametes, eggs or sperm, or spores. Eukaryotic cell division consists of mitosis, which is division of nucleus, and cytokinesis, division of the cytoplasm. So here is the amoeba splitting into two. Cellular organization of genetic material. So a cell's endowment of DNA is called the genome. So DNA molecules in a cell are packaged into chromosomes, but for prokaryotes it is circular DNA. Eukaryotes will be linear DNA. Every eukaryotic species has a characteristic number of chromosomes in cell nucleus. Somatic or non-reproductive cells normally have two sets of chromosomes. Gametes, which are reproductive cells, like sperm and eggs, usually have half as many chromosomes as somatic cells. Eukaryotic chromosome consists of chromatin. It is complex of DNA and protein that condenses during cell division. So DNA associates with special proteins to form more stable structure called chromosomes. So different proteins in prokaryotes, so chromosomes are actually built different. They look different and not really the same. Chromosomes are found inside nucleus in eukaryotes. Human is 46 chromosomes, 3 pairs, one set of 33 for the egg, one set of 33 for one sperm. Each chromosome contains many genes. Gene is a segment of DNA that is responsible for controlling a trait, actually coding for specific protein. So you have nucleosomes, they look like beads on a string, and then these nucleosomes, they will twirl and twine into a chromatin fiber, which is much more thicker. And then they will loop again into loop domains, and then they will loop as thicker as they can, and then it will become metaphase chromosome. This is where it is visible and is thick. Here is the human female karyotype. You have the human female G bands. You can see there's a dark band and then the light bands, and you know that it is a human female because there is an X, and that X has double, so XX, that is a female. Now is a human male. Mostly the number is the same except for the last one. For the last pair you have one chromosome of X and one chromosome for Y. Phases of the cell cycle consists of the M, and then you have G1, S, and G2. M is what we call the mitotic phase, and we have interphase, which is where the cell doesn't divide. We have interphase, which is consisting of G1, S, and G2. So interphase is the stage where cell grows and copies chromosome for cell division. So this is the cell cycle. Most of the time, the cell will be spending time in interphase, which is G1, S, and G2. And then at the end of the cycle, it will enter mitotic phase. It will form two cells from mitosis. In preparation of cell division, DNA is replicated and chromosome condensed. Each replicated chromosome has two sister chromatids, which separate during cell division. So the centromere is the narrow waist of the replicated chromosome where the two chromatids are most closely attached. So here's a picture of the chromosome. That middle area there, which is a little bit thin, that's where the centromere is. And there are two pairs of chromatids attached together with the same centromere. But at the end of the mitosis, these sister chromatids will separate into two separate chromosomes. Mitosis is conventionally divided into 5 phases: PPMAT, prophase, prometaphase, metaphase, anaphase, and telophase. After telophase, we have cytokinesis. So G2 of interphase, the chromosomes are still in a noodle-like form, haven't condensed yet. In prophase, the chromatin has condensed into chromosomes, but still very long. But in prometaphase, they will achieve full thickness, and then cell nuclear membrane is no longer there, already destroyed, and also the spindle fibers have already been created. Okay, so now we have the pro-metaphase getting ready for metaphase. In metaphase, all of the chromosomes are lined up in the middle of the equator. Metaphase, the daughter chromosomes will be pulled by the spindle fibers into the end of the cell. And at the telophase and cytokinesis, the nuclear membrane will begin to reform. Nucleolus will begin to reform, and the chromosome will unfold again into chromatin, and the cell will actually split into two because there will be a cell membrane forming in between them. In prophase, protein condense, causing chromosomes to become visible. Chromosomes separate, moving to opposite ends of nucleus. Centrosomes start to form framework used to separate two chromatin called Metasis spindle, and nucleolus disappears. Here is chromosome consisting of sister chromatids. Metaphase, nuclear envelope fragments. Chromosome becomes more condensed. Kinetochore is formed at the centromere, and microtubules were attached to the kinetochore. As you can see, attachment of the spindle to the kinetochore. In metaphase, chromosomes align to an axis called metaphase plate. Like this. Anaphase, each centromere will split into two, making the chromatids free. Each chromatid will move towards a pole, cell begins to elongate, caused by microtubules not associated with the kinetochore. Like this. 5 telophase, formation of nuclear membrane and nucleolus. So the chromosome begins to elongate to form a long and thin chromatid. And then the cell will form a cleavage furrow, which is a cellular groove in the cell near the old metaphase plate. In that, in the middle, where the equator is, where they used to assemble in the middle in the metaphase, now it is a cleavage furrow. Cleavage furrow will split the cell into two. Cytokinesis happens where you have the division of the cytoplasm. Cyto means cell, kinesis means cut. Okay, you can see the cleavage furrow forming. And this is an onion root cell. You can see that there is many different processes happening together. Let's see if we can spot where the interface is. Can you spot? Okay. Interface would be any cell that still have the chromosome not condensed. So this might be the interface. Interface. Okay. Now let's see if we can spot the metaphase. Can you see the metaphase? Metaphase will be when the cell's chromosomes are in the middle of the equator of the cell. So here is one, here is one, here is also one. Okay. Can you spot the prophase, where chromosomes are created and they are just floating around and not really pulled to the middle? Can you find it? Okay, here is one. Here is the prophase. What about the anaphase? In anaphase, the chromosomes are pulled to the equator by the spindle fiber. Can you spot them? Okay, here is one. Very clear. You can see the chromosomes are being pulled to one pole and the other pulled to another pole. Let's move along. Okay, so we look at the mitotic spindle. It is microtubules that controls chromosome movement during mitosis. So imagine that mitotic spindle is similar to a bungee cord. Okay, attached to you, and then it pulls you to the direction that it wants. So think about spindle fiber is that this during non-mitosis it doesn't exist, but during mitosis or meiosis suddenly it appears. Assembly begins in the centrosome. Microtubule is the organizer center. Centrosome replicates, forming two centrosomes. Get two opposite center of the cell, as when the molecule go out from them. So you would also have an ester. Ester in the Greek term means star. So an ester is a radial array of short microtubules. Stand for each centrosome. So this centrosome, it is at the end of each cell, and from it, the spindle fiber will grow, and then near it there will be like short microtubules that form like a star shape around them. Spindle includes the Centrosome, Spindle Microtubules, and the ester. Some Spindle Microtubules attach to the chromosomes and move the chromosome to Metaphase plane. So as you can see here, this is the Centrosome. Okay, they are at the end of the cell. So they are made out of microtubules but kind of short. And then you have the ester coming out. Ester is kind of like a burst, like a star shape like this. Okay, coming out from the middle. And then there is a lot of microtubules. Okay, where do they go? Where do they attach? They attach on the cytochromatid kinetochores. Okay, and so they will actually be the one that pulls the chromosome through here. The attachment point is kinetochores. In anaphase, cytochromatids separate and move along the kinetochore microtubules towards the opposite side of the cells. The microtubules shorten by depolymerizing at the kinetochore ends. What does it mean? It means that at the end where it attaches with the kinetochore here, it will slowly disintegrate. So imagine like if you are pulling something, right? You're pulling something with the rope. As you pull, you cut the rope behind you. So as you are moving towards the thing that you're pulling, you also cut the rope behind you, meaning that the rope actually gets shorter and shorter as you are closer to the thing. So this is how it looks like. You have the motor protein. Motor protein would kind of slide along the microtubule, but at the end of that motor protein, the microtubule would be broken down into tubulin subunits. One kinetochore microtubules from poles overlap and push against each other, elongating the cell. As you can see in this picture, which are the non-kinetochore microtubules? Are they the red one or are they the blue one? They are the blue one because they are not attached to the kinetochore. That's what we call it, the non-kinetochore microtubules. So they push against each other to elongate the cell. In telophase, genetically identical daughter nuclei form at opposite end of the cell. In animal cells, Oogenesis occurs by a process known as cleavage, forming a cleavage furrow. In plant cells, cell plates form during Centromere. As you can see, this is the cleavage furrow. This is the cleavage furrow. Cleavage furrow will be the middle part where the cell is dividing into two. Okay, and so the cleavage furrow is actually made by microfilaments which are contracting. Microfilament, when they contract, will actually cause the area where it contracts to be a little bit narrower and narrower. So imagine like you are tying a rope around a bottle and then you pull against that rope. So what happens is that the middle of the bottle will be narrow and narrow. So at the end of this cytokinesis, this cleavage furrow will make the cell split into two, and then it will become two daughter cells. Okay. So here is the mitosis of an animal cell. How can you tell whether it's an animal cell? It doesn't have a cell wall. Okay. So you have interphase, I have prophase, metaphase in the middle, anaphase, they are being pulled to the end. Early telophase means that you can see that in between the cells there is something forming in between. So this is the cleavage furrow. Then at the late telophase, now we have two cells with a wall in between. Okay, what about plants? So plants, they do not have a cleavage furrow. Instead, they will actually build a wall using cell plate. So there will be some vesicles forming in between the cells during anaphase, and then this cell plate will combine together to make a new cell wall. So let's see from number one, prophase, chromatin is condensing, Prokaryotes disappear. Okay, and then mitotic spindle is starting to form. And then from metaphase, you can see the chromosomes have been formed. We have two sister chromatids, and then in the metaphase, all of the chromosomes has lined together in the middle. In anaphase, all of the chromosomes are being pulled to the poles. At the telophase, you can see that the chromosomes are reforming to chromatin, and in between there is a cell plate being formed. And now for meiosis. Meiosis is a reduction of genetic content and chromosomes. So it reduces number of genetic material by half, produces haploid gametes and spores, each containing one member of homologous pair of chromosomes. So in meiosis, there is a certain event that is unique only to meiosis. It is called crossing over. In crossing over, the chromosome pairs will exchange their genetic material between members of homologous pair chromosomes, meaning that they can actually shuffle their genetic information between each other. So meiosis 1 is called reductional division, meaning that from 4 to 2, from 2 to 1, whatever chromosome that you have, it will be halved. Meiosis 2, you have equational division, meaning that you have the number of chromosomes, and then the next what will happen will be the number of chromosomes will be still the same, it's just that you now have sister chromosomes. So DNA synthesis occurs only once during interphase before the beginning of meiosis 1. It does not occur again before meiosis 2. In meiosis and in mitosis, there are differences. Mitosis, the division happens only once, and then you have daughter cell and another daughter cell. Meiosis 1, after the first division, there's another division. Okay. So in mitosis, in this organism, you start having 4 chromosomes. After mitosis, you have 2 cells with also 4 chromosomes. For meiosis, you start with 4 chromosomes, but in the end of meiosis 1, you have 2 cells with 2 chromosomes. In fact, it's just half. In meiosis 2, you have another 2 cells coming out from these 2 cells, forming 4 cells with each 2 chromosomes, which is half of the original. So the first meiotic phase 1, okay, you have prophase, metaphase, anaphase, and telophase, similar to meiosis. Okay, so meiotic phase 1, you have euthymema, zygodyma, pachynyma, leukemia, and gametes. In euthymema, the chromosomes are so thin, and in zygodyma, the chromosomes form a bivalent, meaning that 2 homologous chromosomes, they are close to each other, will form couples or pairs. And then each one of these chromosomes could double to form sister chromosomes by themselves. Okay, in pachynyma, that's what happens. You have now 4 sister chromosomes. Okay, each one of the chromosomes, they form sister, so now 1, 2, 3, 4. Tetra. Okay, tetra is 4 in Greek. And then you have diplonema. So in diplonema, the chromosome will form a chiasma. The chiasma is the place where the crossover happens. And then in diakinesis, you have terminalization. After chiasma, they have exchanged their segments, and now the actual crossover is complete. Metaphase 1, anaphase 1, telophase 1, and prophase 1. Okay, so as you can see here, the chromosomes from 4 now becomes 2 for each chromosome. And then metaphase 2, anaphase 2, telophase 2, and lastly the cytokinesis 2 again. Okay, so as you can see, from 2 chromosomes, it becomes 2 chromosomes. It's just that each one of the sister chromatids, they split. So prophase 1, the first meiotic division. There are 5 substages: lepto, zygo, pachy, diplo, and diakinesis. And the end of prophase 1, we have Tumors present on the equatorial plane. So leptonema, long sickle tracts. Zygodyma, synapses. Pachynyma, shorter, thicker, and then you have chromatids, sister chromatids called tetrads, and crossing over. Diplonema, tetrads, sister chromatids separate, except for the chiasma. Diakinesis, nuclear envelope, and nucleus breakdown. Centromere is attached to spindle fibers. Metaphase 1, we have the collage, maximal shortness, terminal chiasma, while holding no sister chromatids together. Minor spindle fibers move from the metaphase plane. So in between the sister chromatids, we have cohesin. Cohesin is a protein that keeps the sister chromatids together. So during anaphase 1, cohesin is degraded. Means the sister chromatids will now be separated. Homologous chromosome will separate, and no junction 1 occur. Telophase 1, the membrane forms, and nucleus enters interphase. So, what is summary of MAT1? MAT1, yes, homologous chromosomes separate most of the pole, sister chromatids may attach. Regular chromosome will reach the poles. Each pole now has haploid chromosome. In cytokinesis, you have 2 haploid daughter cells formed, and nuclear envelope will form around the chromosome in some species. And then what you can see here is the crossing over. In the crossing over, chromosome will exchange the genetic material, so it will increase the genetic variation. Prophase 2, Metaphase 2, Anaphase 2, and Prophase 2. In the second meiotic division, Centromere are separated into anaphase poles in Anaphase 2. Each haploid cell from Meiosis 2 now has one member of each pair of chromosomes. So as you can see, these two chromosomes, 1 and 2, now they are split into 6 chromatids and they end up as 2 daughter cells. So these 2 daughter cells also come from 2 daughter cells, so now we have 4 daughter cells. Development of gametes varies in spermatogenesis compared to oogenesis. 2.5. The development of gametes varies in spermatogenesis compared to oogenesis. Okay, so what we have learned is that meiosis will create 4 cells, 4 daughter cells, right, from 1 to 4. Okay, so male gametes, meiosis happens in the sperm. So spermatogenesis is the meiosis for the male sex cells. For females, it is oogenesis, the creation of ovum in the ovary. So if you want to compare, in spermatogenesis, one spermatogonium will create 4 secondary spermatocytes. In spermatogenesis in males, one spermatogonium will create 4 spermatozoa. So it is similar to the meiosis that we have just learned. But for oogenesis, one oogonium will only create 1 ovum. So what happens to the other 3? Okay, so in oogonium will turn into a primary oocyte. So a primary oocyte will divide into a secondary oocyte, 2 of them, but 1 of them is lost as a first polar body, meaning that only 1 survived. So this secondary oocyte, which has survived, it will divide into 2 cells. Only 1 cell is destined to become the egg cell. The other will become the second polar body. So surviving cell is called an oocyte. So this oocyte will differentiate to become an ovum. So you can say that primary spermatocyte undergoes meiosis 1, produces 2 secondary spermatocytes. Spermatocyte undergoes meiosis 2 to create 4 haploid spermatids. So you can see here in these 3 pictures, testis mass as a percentage of body mass. So Prokaryotes, 13.8% of the body mass is the testis. Fruit fly, 10% of their body mass is testis. So oogenesis is different. Out of the 4 cells that might form, you only have 1 ovum. The other 3 becomes polar body. Meaning that in this process, in the 4 daughter cells, 3 does not receive the cytoplasm similar to the one that is destined to become an ovum. But the 1 daughter cell receives cytoplasm, it is the primary polar body. It undergoes meiosis 1 and 2, it develops into the ovum. So, meiosis is critical to the successful sexual reproduction of all diploid organisms. Meiosis is a mechanism in which the diploid number of genetic is halved. So fungi, haploid representative cell arise during meiosis. So fungi proliferate in the mitotic cell division. For plants, life cycle alternates between sporophyte stage and metaphase stage. Okay, so the fertilization of the seeds. Okay, within the seeds, you have the egg and sperm. If you look at flowers, the actual sperm of the plants are actually the pollen. The pollen are actually macrospores or the male sex cells of the flower. Okay, and then the female sex cells of the plant are called ovules. So ovules and anthers or pollens are actually on the flower. So both of them, if they fertilize, they will actually form a seed, which is called zygotes, and then it will form a sporophyte, which is the actual plant. So what about the prokaryotes? Prokaryotes do binary fission. In binary fission, chromosome replicates and the two protein chromosomes actively move apart. Chromosome replication begins in the beginning, and soon after, one copy of the origin moves rapidly toward the other end of the cell. Then replication continues. One copy of the origin is not at each end of the cell. And then replication finishes, the plasma membrane goes inward, and new cell wall is deposited, creating new daughter cells. So the evolution of mitosis. Since prokaryotes evolved before eukaryotes, mitosis probably evolved from binary fission. Certain proteins exhibit a type of cell division that is intermediate between binary fission and mitosis. In this type of cells, they're kind of like in between. Prokaryotes, you can say that they are binary fission. The DNA is split to two, then becomes two DNA, and then becomes two cells, right? For the non-flagellate, the nuclear envelope are still there, they are not destroyed. But microtubules, they are outside. The red algae, they have Kinetochore microtubules and an intact nuclear envelope. And then eukaryotes, there is no nuclear envelope at all. And then the Kinetochore microtubules are even more than the previous example that I've shown. Cell cycle control system. So the cell itself obeys some kind of clock. Okay, so the clock has specific checkpoints where the cell stops until the go-ahead signal is received. For many cells, G1 checkpoint seems to be the most important one. So you have a G1 checkpoint, G2 checkpoint, M, and then back to G1. So we have 3 checkpoints. If in the checkpoints the requirement is not received or is not met, then the cell cannot progress to the next stage. So at G1 checkpoint, if the cell receives a go-ahead signal at the G1 checkpoint, cell continues on in the cell cycle. But if a cell does not receive any go-ahead signal, it will exit cell cycle and go into 0. G0 is a non-dividing state. Okay, so what are the molecular mechanisms that help regulate cell cycle? It is called cyclin. Okay, so if there is a lot of cyclin, okay, when they accumulate, it will become an MPF, mitosis reproduction factor, and then during mitosis, cyclin is degraded. Then when it degrades, CDK will be left. So as the cell goes on, we will accumulate cyclin as it lives, and then if cyclin is enough to form MPF, then mitosis will happen again. Okay. An example of an internal signal is that kinetochores not attached to a spindle microtubule will send a molecular signal that delays anaphase. Some external signals are growth factors, so protein released by certain cells that simulate other cells to divide. For example, you have PDGF. PDGF is a platelet-derived growth factor. It simulates human fibroblast cell division in culture. Okay, so you put some cells and then you try to make it divide. Okay, so without PDGF, you just stay. You will just not divide because there is nothing telling them to divide. But with PDGF, this growth factor, the cell will actually be stimulated to grow and divide. Another example of external signal is density-dependent inhibition, which cardiac cells start dividing. So most animal cells exhibit encouraged dependence. They must be attached to a substrate in order to divide. So they have to anchor on something. So cells anchor to this surface and divide. So we have anchoring dependence. When the cells have formed a complete single layer, they stop dividing. This is called density-dependent inhibition. But if you scrape some cell away, the remaining cells divide to fill the gap and then stop. So it is also density-dependent inhibition. So these are the normal mammalian cells. But for cancer cells disobey this inhibition. Okay, so they do not have any encouraged dependence. They don't have any density-dependent inhibition. If they actually hit a wall, they still will continue dividing. And if they get scrapped off, they will also keep dividing. So meaning that they do not need a substrate to divide. They don't need to anchor. They can just divide however they want. In cancer cells, there is a loss of cell cycle controls. Usually for regular cells, you have a cycle of not growing and then growing. Okay, you have G1, as G2 and then M. But for cancer cells, it's always M. Always divide. Okay. So cancer cells form tumors, massive or abnormal cells within otherwise normal tissue. If abnormal cells remain at the original site, the lump is called a benign tumor. But if you have malignant tumors invade surrounding tissues and can metastasize, exporting cancer cells to other parts of the body where they may form secondary tumors.